Welcome to the 5th Stanford Inventor webinar. We're pleased to have Dr. Noah Burns with us today. I'm Ann Copsill. I'm the Licensing Associate at the Office of Technology Licensing here at Stanford. We'll take questions at the end. Dr. Burns will talk for about 20 or 25 minutes. Please use the chat function on your webinar screen and go ahead and ask the questions at any time, and we'll queue them up to ask at the end. And you may either ask them anonymously or publicly. So Dr. Noah Burns got his bachelor's degree in chemistry from Columbia. Then he went to Scripps Research Institute for his PhD and did postdoctoral work at Harvard. He came to Stanford as a faculty member in 2012. And he's here today to talk to us about a scalable synthetic process for creating laterane phospholipids.
Thanks, Ann. Good morning, everyone. I am very excited to have this opportunity to tell you about this particular project that we've been working on in my lab for a number of years. I'll first give just a brief overview of the kind of work that we do in my lab. So I am a synthetic organic chemist, and we mostly look at natural products, so interesting small molecules that are found in nature with a variety of potential properties and activities and hopefully applications. The other thing that we focus on in my lab is developing new organic methodology for making small molecules. The natural products that we've worked on in my lab kind of fall into two camps. So one are small organohalogenated natural products, ones with chlorine and bromine. There, it's sort of it's more biological activity that we're interested in and following up on. This project is related to the other arm of our research, which has to do with unusual, as we call them, non-canonical membrane lipids. And we are interested in developing processes to make them, but we don't like to stop there. We are trying to find applications of these. That's what part of this is about, obviously. And I will today talk about, I'll give a little background on the molecules themselves. I'll talk a little bit about the synthetic process that we've developed. And then I'll talk about some of the properties that we've been discovering as we're testing and doing some sort of standard biophysical type experiments. So this is the molecule, a representative molecule that has to do with this work. So these are, or this is one of the so-called laterane phospholipids. These are highly, highly unusual small molecules. I think anybody that looks at these with any kind of organic chemistry background is shocked by the fact that there's not one, but multiple cyclobutanes that are fused. And of course, that's going to mean there's a lot of strain in these compounds. And that also means that they have been a pretty big synthetic challenge to attempt or to accomplish. And again, that's something that we've been working on in my lab over basically since we started in 2012. So there are two motifs, as we call them, of sort of laterane motifs in these laterane phospholipids. There's a so-called 5-laterane, which is 5 cyclobutanes fused to one another. There's also the 3-laterane, as you see here, with 3 cyclobutanes fused to one another. This is a representative phosphatidylcholine, so it has a phosphatidylcholine head group. But of course, in nature, and I'll talk about where these come from and some of the interesting sort of background and questions surrounding them. But there's a huge diversity, right? Just like in any living organism, there's going to be a huge diversity of lipids. And we wanted to develop a controllable synthesis so we could make any of these. And again, these are the only naturally occurring polycyclobutane motifs, but our synthesis would allow for making any number of cyclobutanes fused to one another that we might desire. So that's one of the beauties of synthetic organic chemistry, right? We're not limited by just what nature's making. That's where we start, but then we go on to make non-natural compounds too, and I'll talk about just one of those briefly today. Okay, so I want to give a little background on these molecules. It's just kind of an interesting story, but it really ties into the things that we've been trying to do and study. So again, these laterane phospholipids, they come from a bacterial species, and it's estimated that up to 60% of all the lipids in these bacteria contain some polycyclobutanes. That's a low estimate. I think it's even more. So the ubiquity of these things in this organism has said to us that there are some really interesting purposes for why nature has decided to make them. I have some quotes here from the literature that are basically saying we cannot get enough of these materials from nature in order to do the studies. These are related to biophysical studies, which when we started in 2012 was a really nice justification for us for trying to attempt to do this. It also helped us get some funding for this research, which is obviously very important. Okay, so 2 more slides on background here. So the producing organism for these compounds are so-called anammox bacteria. So that stands for anaerobic ammonium oxidizing bacteria. You'll see this equation in the middle of the slide, and that is the primary source of energy for these bacteria. This is this so-called anammox process, and what it does is it couples ammonium NH4 plus with nitrite NO2 minus to make dinitrogen and water. That's thermodynamically downhill, and again, that is their main source of energy. Okay, it's now estimated up to half of all the nitrogen produced in the ocean comes from these bacteria. So we like to say they are globally important bacteria for the nitrogen cycle. Now, based on the ubiquity of these and the fact that you can culture these in the lab, it's not so easy. There's only a few people in the world that have been able to develop these anammox bioreactors. But you can culture them, and as you see from the 50% of nitrogen that's produced by them, they are ubiquitous in nature. But the difficulty in actually getting them pure has to do with the fact that there are a lot of very similar structures. So separation has just proven to be a really big problem, and hopefully that the quotes on the last slide help sort of accentuate that. Okay, so here on the left of this slide is a TEM image of an electron microscopy image of one of these anammox bacteria. And what I want to point out is there's a very large intracellular compartment, so that's the lighter space within the cell, and that is the so-called anammoxosome. You can see it in the cartoon picture, sort of in the right-hand picture, and that is the power plant of the cell. It's like the mitochondria, if you will, and that's where that anammox process of ammonium plus nitrite to nitrogen and water occurs. And the reigning hypothesis for what these molecules do, or what one of the purposes of them is that they, and this was proposed by the original isolation chemists, they proposed that these lipids pack in a very dense manner and make a bilayer that is impermeable to small molecules. Okay, and the small molecule that they were talking about are some of the intermediates in the anammox process. So if you see in the sort of catalytic cycle through the membrane bilayer in that cartoon, some of the intermediates are hydrazine, so N2H4, or hydroxylamine. These things are highly toxic. They're toxic to any living organism. So the idea, and I think it was, it's certainly an interesting one, was that, again, these laterine lipids pack very densely and protect the rest of the cell from diffusion of this hydrazine out. Okay, now, I hope to be able to show you that this hypothesis is not actually correct, but I don't want that to undermine the fact that these bilayers are quite impermeable to certain small molecules. I think hydrazine is kind of an exception. It's very small, but we are finding, and other people have found too, you can see this in the literature, that small molecules do not diffuse very well across these bilayers. Okay, so we want to address function and potential applications. This slide summarizes our synthesis of the 5 laterane, so that's the 5 cyclobutanes fused to one another. I'll point out just a few key reactions. The very top middle reaction that uses KOTBU, so potassium terbutoxide, that is an abnormal olefination or alkene synthesis that we have sort of developed, and we found it to be uniquely effective for making strained olefins. And these cyclobutenes are highly strained, so this has been a really nice way to make this. We can make that compound on as large a scale as we want. So I have 15 grams per batch on this slide, but we could do it on even larger scale. Now, this is not a new compound. It's been known in the literature, but this process we've developed is much more efficient than the ones out there. We then do a photochemical 2+2 to make the compound in the top right, which I have an X-ray structure of. That is a unique reaction that works on non-activated olefins. So there are very few ways of bringing together 2 alkenes that are not electronically activated in order to make cyclobutanes. And so we've developed a process there, and it really was sort of an engineering feat to get that to work, because we have to do it at low temperature, but it is at this point fairly scalable. We then have to do some CH activation, so-called CH activation chemistry, in order to functionalize this hydrocarbon and then extend the side chain. And our synthesis is flexible. Hopefully you can see in that we can attach any length side chain that we want, and this is an enantioselective synthesis, so we are getting just 1 enantiomer, or predominantly 1 enantiomer out. And so we have this synthesis of this so-called 5-lateranoic acid, as you see here. I say we've made 600 mgs of this. I'm sure at this point it's well over multiple grams. And I do think this is a scalable process, as we say in the title. And I would compare this, I'll have the summary of the 3 laterane synthesis on the next slide, but so I remember it now. Remember to say this, the price of this, we estimate the price of this process to be very similar to other natural product syntheses that people have done and that have been published in the literature by both academic and pharmaceutical companies. So here's the synthesis of the 3 laterane. Hopefully you can see that the bicyclohexene that we made on the previous slide is also one of the key components here. So we construct the whole hydrocarbon carbon core in the top right. So you see it has the 3 cyclobutanes on the cyclohexane. We have a handle in that bromine in order to install a side chain. So again, we are very flexible in what side chains we can make or install there. And then we have to reduce everything down. So I like to say that one of the challenges in these molecules is that they have no functional groups, right? They're just hydrocarbons. And so we use these functional groups to make the core, and then we have to burn everything down, if you will, just reduce everything. And so the unique solution that we've come up with and invented here is to take this alkene diketone and then condense condense hydrazine onto both of the ketones to make this alkene bis hydrazone in the bottom right. And then that reaction, the copper-mediated reaction, that's sort of the main invention for this 3 laterane synthesis. And we call that an oxidative isomerization. So we lose 2 equivalents of N2 and convert the ene bis hydrazone into this cyclohexadiene. And then we can hydrogenate, that works fine under standard conditions, and then deprotect the protecting group of the alcohol. So I have the 3 lateranol here. We can also make the acid. We could also make the alcohol in the 5 laterane synthesis. And again, same thing goes for this. We've made multiple grams of this. We can essentially make as much as we would want in our lab and as much as we've needed in order to do some of the follow-up studies. Okay. So this is just to sort of summarize the head group installation. So this is known literature chemistry for making phosphatidylcholine. So we have this functionalized glycerol, and then again through literature process, we have another way that we've done this too, which is slightly more efficient. And the phosphatidylcholines, the laterane phosphatidylcholines that we've made so far are on the bottom of this slide. So the top 2 are both naturally occurring. So we made these and we've studied their properties. The bottom compound is non-natural. Okay, just again to sort of show that our synthesis is flexible. We can make anything that we want. We're not limited by what has been found in nature. Okay, so one of the first things we needed to do was just measure melting temperatures and make sure that these compounds actually do self-assemble into vesicles. One of the people we've collaborated with on this project is a professor named Steve Boxer. He's in the chemistry department, and he's really a world's expert on lipid biophysics. And when he looked at these compounds, he was not sure that they would self-assemble because, right, if you compare these to normal straight-chain phospholipids, they're much wider. So he was thinking, that could really disrupt their packing. It turns out that they actually do the opposite, and I'll be showing that with some of our data here. Okay, in the top, we have these 2 natural laterane phosphatidylcholines, the 3,3 as we call it, and the 5,3 PC. Those melt, below room temperature. So if we add a little bit of lipophilic dye, they self-assemble and the dye goes into the bilayer, and then we can visualize it through these fluorescence microscopy images. The compound at the bottom, this non-natural compound, melts at a much higher temperature. And so if we heat this up in water with the dye and then cool it down to room temperature, we see this sort of phase separation. So that's indicative of being in the gel phase at room temperature. And we have other data to support that. Okay. Here's a study that we've done to measure lateral diffusion. So this is a standard technique called fluorescence recovery after photo bleaching or FRAP. And the way this works is we deposit a bilayer onto a surface and then have a dye in that we can photo bleach. And so we hit it with a laser. The top left image is supposed to be sort of a little movie of that. It may not be coming through, but it's not that important. So we bleach a spot with a laser and then watch the fluorescence watch the dye diffuse back into that bleached spot. And that again just allows us to measure lateral diffusion within the bilayer. And so the bottom left graph shows this. So DOPC, standard canonical membrane lipid. When we photobleach that at T0, you see the fluorescence goes down and then pretty quickly recovers. With the natural 5,3 and the 3,3 PC shown in the red and green lines, hopefully what you see is that recovery is much slower, meaning that lateral diffusion is also much slower. The 5,5 PC shown in red, we know that's in the gel phase. So if we photobleach that, there's no diffusion as would be expected. So one of the things that this slow lateral diffusion is potentially saying, and like the data on the next slide, it corroborates this, is that there are much, much stronger intermolecular interactions between laterane hydrophobic tails, if you will, or between those polycyclobutanes. And we think that this could absolutely be one of the reasons that they are being made in nature. My colleague Steve Boxer likes to say that these lipids kind of exist in a unique phase and not your sort of normal totally fluid phase, but they're definitely not in the gel phase, at least the natural ones. So this has been very interesting for us to investigate some more. We've done a Langmuir monolayer isotherm analysis. I know there's a lot of data on this slide. What this is hopefully showing is that these laterane lipids are not as compressible. So they, again, are packing very densely when we apply force to a monolayer that's put down on a water surface. What we see is essentially no resistance until they self-assemble to their sort of packed place and don't really push back that much. One of the really interesting things that we found from this Langmuir data is that if you look at one area on a monolayer, bilayer composed of these compounds, as I said, they're wider, so our expectation was that fewer of them would exist within a particular area as compared to normal straight chain phospholipids, but this is absolutely not true. So it's the opposite. It actually turns out that more of these wider molecules pack into one area than your canonical straight chain lipids. Again, really pointing to some very strong intermolecular interactions between hydrocarbons. And that actually has been corroborated by some computational evidence. There's one paper out there where people computed interactions between polycyclobutanes and actually predicted that they have some of the strongest van der Waals interactions, intermolecular interactions, which I think is pretty interesting and clearly related to their function. Okay, we've measured bilayer thickness using standard small angle X-ray scattering up at the Stanford Linear Accelerator. And what I hope that you can take from this, so the two peaks, right? So this is a map of electron density across the bilayer, and the two peaks that you see are the two phosphatidylcholine head groups, right? That's where there's a lot of electron density, so you see that. So peak to peak is the bilayer thickness. There's nothing that surprising about the thickness, but what is surprising is that the electron density within the bilayer is much higher than the canonical lipids. So those two lines that just go straight across in the middle, those are our natural laterane phosphatidylcholines. And again, so showing that the electron density is much higher there, which is also indicative of much tighter packing within the bilayer. So to sort of summarize the properties that we found so far, we have found that these laterane lipids pack they do self-assemble like normal phospholipids, but they pack much more densely and make much more viscous bilayers. We wanted to measure hydrazine diffusion, so we developed an assay for doing that. And what I want you to take home from this, looking at the data on the left, is that what we've actually found is that these laterane phosphatidylcholines are not particularly impermeable to hydrazine. I know this is kind of it's a negative result, but we think it's overturning the hypothesis that's existed in the literature. And I think, I'm not so sure that there's potential applications based on the hydrazine, but again, we just wanted to see this. One of the most significant results we've gotten so far has to do with proton impermeability. So this is a known assay for measuring pH equilibration into or out of a vesicle. And in the data table, hopefully you'll see the half-lives of the canonical straight chain lipids in seconds of proton diffusion. And at the bottom of this slide, at the bottom of the table, I want people to see that the proton diffusion, excuse me, equilibration across a bilayer composed of lateranes is an order of magnitude slower than canonical lipids. And we think that this could absolutely be tied to the function of the natural function. Regardless of that, it's a very interesting property. And as far as we're aware, these are the least permeable phospholipids to proton equilibration of any that have ever been discovered. So a very unique property. And we do think that there could be potential applications just based on this. So for studying potentially membrane proteins, so there are membrane proteins where one might want to study them and actually maintain a pH gradient over longer periods of time. So this could be useful for that. We think they also could be useful for protein crystallography, so membrane protein crystallography. right? There's a lot of membrane proteins out there that we've yet to really understand because we can't crystallize them. And we think that these lipids, which exist in this sort of unique, right, intermediary phase between gel and fluid, could help with that in that, diffusion and hopefully self-assembly of these membrane proteins would be faster. Okay. So this is the final slide I have, scientific slide. There's a little data. There's a statement at the bottom that says that the 3,3-phosphatidylcholine leaks this water-soluble dye about 2 times slower than a conventional membrane bilayer or vesicle composed of DOPC. Now, this is a little misleading, I think, because for greasier compounds, so maybe more drug-like molecules, we think that diffusion of the drug out of the vesicles will be much, much slower. And we haven't measured that yet. We think that would be pretty easy to do. But we think that could be a very unique property and one that could have applications in liposomal drug delivery. So we have done one study with looking at drug delivery. I don't have any of the data shown here. In that study, we did show that, or we did test the lateran lipids against some normal human cell lines and didn't see any toxicity. And we did see a very nice encapsulation of a doxorubicin anti-cancer drug, just as sort of a proof of principle there. So that's pretty much all I wanted to share with you today. I would be more than happy to answer any questions if anybody has them. Thank you.
Great. Thank you, Dr. Burns. And let me tell the audience that if you have questions, there's a chat function. Please ask your questions there and they'll come into our queue. We have a few here already. Actually, I think you talked about this maybe right at the end, but the question had come in about toxicity, and especially for if people wanted to use it for human therapeutics.
Yeah. So again, we don't have a lot of data on this, but in some normal, I want to say they tested it on human liver cells, and there was no measured toxicity. I don't know exactly how high a dose we went. I could absolutely get that data if that would be helpful to anybody.
Great. And you talked a little bit about the cost and that you had scaled it up to grams, but can you talk any about the ability to use it kind of commercially, the cost people might expect, and the ability to scale it up in a...
Yeah, so I mean, I will have a hard time giving true numbers here. I think let me say that in my lab, in an academic lab, we are at the sort of, I would say gram scale limit as far as the final lateran lipids are concerned. I do think given money and resources, I think scaling it up would be fairly easy. But I really think it would be the infrastructure that would be probably the biggest thing. That one photochemical reaction that I mentioned, that copper catalyzed photochemical reaction, requires a little bit of special engineering. But I mean, I'm sorry. I know that maybe isn't the most satisfying answer to not have a true number attached to it. Yeah, I think that's all I can say on that. I mean, I do want to emphasize it's not different from other I don't think it's any different from other natural product syntheses that have been done in academic labs. That being said, those are rarely scaled up to kilogram scale. That there are some really beautiful examples of that, but they're a little more rare. So I do think we'd be, in my lab at least, limited more to gram scale access.
So we've got a few more questions here. One, is there a definite plan to conduct the drug delivery capability? And if so, what will you test?
So we currently, we do not have any more concrete plans to do that. One thing that we are planning to look at is not the strict natural vesicles themselves, but looking at polymers that have these lateran tails attached to them. So there are certain polymers that have been used for drug delivery, and sort of common type polymers used for drug delivery. And we are planning to do those. And we do actually have some preliminary data that shows that they hold on to Taxol better than the normal straight chain greasy, straight chain side chain on these polymers.
Great, and here's another question. Have you measured the initial size and lamellarity of liposomes that form when the material is hydrated? Have you then tested the ability to reduce size by sonication or extrusion?
Absolutely, that's a great question, and we have absolutely done that. So sonicating aqueous solutions of these, we make GUVs of the tens of micrometers. We can make them smaller and more unilamellar through normal extrusion. So those normal techniques, absolutely, those give us small unilamellar vesicles, and those are actually, so the SUVs are what we've done all of the permeability studies with. Those images I showed are GUVs, just to show, easier to see things that are bigger. But there's sort of nothing surprising there. I would say they've behaved for us in the same way that your standard canonical membrane lipids would. So nothing special there.
An additional question from there. Do you have any data showing protein reconstitution in Laterane's membranes?
We don't. We haven't done that yet. But that is one thing that we do have plans to do with Steve Boxer, with Steve Boxer's lab in our department. But unfortunately, we haven't done that yet.
Let's see. And here's one. I could take this. What's the status of patenting and licensing? So there's been a PCT application filed in August of 2017. So that's published, if people want to look that up. It was published a few months ago. It's got Laterane in the title, and of course, Dr. Noah Burns is one of the inventors. And the technology is available for licensing. You contact me, which I think, is on the screen. Great. And let me ask, are there any more questions from the audience? If not, great. I think we'll wrap it up. Thank you very much, Dr.
Sure.
Burns.
Thank you, Ann. Thank you, everyone.